tables, but this one I want to show you is outlined in bright red. So I want you to know that these terms are on the exam. There's a couple of questions. We're familiar with aseptic, right? We're minimizing contamination. Disinfection and antiseptics, that's the same thing. The only difference, right? We're reducing microbes. The only difference is going to be where. Antiseptics is on tissues, on the skin. Disinfectants are on inanimate objects like the tables, doorknobs, right? You're going to obviously use different stuff. You're not going to spray the stuff you clean on tables with on yourself. Sterilization, completely free of microbes. De-germing is like scrubbing, physical removal. Pasteurization we're familiar with, right? For food, we're decreasing microbial load. And then two suffixes, side, so bactericidal, homicidal, right? You're killing. Bacteriostatic or microbiostatic or fungistatic. This is going to be where you're decreasing or basically inhibiting reproduction. So we refrigerate our food. That's going to be an example of a microbiostatic, whereas microbicidal or bactericidal, you cook your food. Okay, so this just shows death rate. Keep in mind, even if you use like, you know those little hand sanitizers that say 99.9% of bacteria, well, if you take into account the sheer number that there are, 99% of that is not everything. You still have a considerable amount. If you're thinking like trillions and trillions of bacteria, well, 90% of that, you still have a lot left. So, yeah, it's not immediate. It takes some time, right? Okay, so common theme for both of these are, if you disrupt the cell membrane, the cell dies. If you disrupt the cell wall protects it against osmotic effects. So remember, our red blood cells, they can explode, they can shrink because they're not protected by a wall. So the same thing, when you damage their cell wall, they're now susceptible to osmotic forces. This bottom part, I'll come back to whenever we talk about viruses for chapter 13, so I'm not going to spend any time on it right now. You damage proteins, you damage DNA, the cell dies, ultimately. Okay. Alright, so ideally these things should be not super expensive, they should be relatively fast, stable during storage, and then the most important part, I think, is harmless to us, right? You don't want to be using something on the table to clean it that will eventually give you cancer. So the big thing, or the big theme for both of these chapters is harmless to humans. Okay. I do want to spend a little tiny bit of time on this. So most susceptible to most resistant. So most bacteria are generally susceptible, especially if they're actively dividing. Then once we get into more hardier stuff, things that are harder to kill, mycobacterium, right? We talked about tuberculosis, we talked about leprosy, right? So these guys, by their very nature, because of their mycolic acid, are very hard to kill. Cysts are kind of like the endospore equivalent for protozoans, but endospores. So if you've ever been in a medical environment where they use an autoclave, they have the little spore tape. I have a picture of it later on that we'll look at. This indicates that you've met the conditions to kill these guys. That's all it indicates. We'll talk more about that in a second. Prions, these are pretty scary. We'll talk about that for chapter 13. Okay, so obviously we can classify these germicides based on how effective they are. High level kills everything, including your spores, intermediate, some stuff, and then low level is just going to be most of your vegetative bacteria, certain viruses, all that stuff. Temperature and pH affect stuff. Organic material why we wash our hands. That's why we're using lab coats. So if you're healthy, exposure is not a big deal. If you're immunocompromised, if you handle it safely, it's still not a big deal. You'll be okay. BSL-3, this is where we start to get into more serious stuff that exposure and improper handling can give you some disease that is pretty intense. So we've added like little safety cabinets, which is kind of similar to what that was when we did our spore stains. HEPA filters. BSL-4, that's like, even if you handle it properly, minimum exposure is going to kill you. It's not even like, yeah. So you have to wear the space suits, the safety cabinets, the HEPA filter, the airlock doors. So Ebola, smallpox, where not only will it get you sick, you also have the chance of producing a small pandemic, right? There is a BSL-4 lab in Galveston, so if you're hoping that a hurricane doesn't knock it down. Okay. Physical methods, you're messing with the environment, whether it's temperature, salt, all that stuff. So, high temperatures denature proteins, disrupt the cell membrane, disrupt the DNA, you die, right? So those are cidal methods. Alright, so different types of heat-related methods. Moist heat, boiling is an example, autoclaving, pasteurization. So boiling, obviously, boiling does not sterilize stuff. It kills stuff that gets sick, right? But endospores, cysts, certain things can survive boiling. Boiling is not sterilizing. Autoclaving, this is what they use in a lot of medical offices. So it's pressure and boiling kind of coupled together. These are those spore strips that I was talking about. So they'll put that little piece of tape to indicate that the conditions to kill an endospore have been met. More accurately is this. So this is what we've seen. This is phenol red, so a pH indicator. And that little vial contains food as well as that pH indicator. On the bottom, you have a little spore strip, meaning you have little endospores on that little piece of paper. Once you've ran your autoclave, you crack that guy open and that releases the food. If your guy is dead, well, it's not going to eat the food, it's dead. But if it's still alive, the pH will change because it's eating the sugar and it'll turn a yellow color. So this is more accurate than tape. The autoclave is supposed to use pressure and heat to kill, and so the tape indicates that the endospore, the conditions to kill endospores have been met, but it's just saying the conditions. It doesn't guarantee that they're dead.
Is there going to be a question like which one is more on the tape or this one is straight?
Oh no, I'm not going to ask you like about the tape or this early. Yeah. I was just explaining. Pasteurization, we've talked about, so remember that's where for food we lower the microbial load to extend shelf life, and then sterilization is the only thing that completely gets rid of everything. So that's why those little packets of dairy creamer, they don't have to be stored in the fridge because they're sterilized. There's no bacteria left in it to spoil it. So there's no reason to put it in the fridge. There's nothing growing. It's all dead. Not going to ask you about times or temperature conditions. Alright, dry heat. You can't use moisture on everything. Some things oxidize, like metals, powders, oils. So dry heat, like incineration. Alright, opposite end of the spectrum, we've got static processes. So you're cooling things down, decreases metabolism, which leads to slow growth, slow reproduction. So that's why we refrigerate our foods, right? So obviously there are some exceptions. So Listeria, right? That's a mesophile that's capable of reproducing in colder environments. Lyophilization, that's where we're removing water, so drying as well as using colder temperatures. Okay, you can also filter these guys out. Keep in mind they're super tiny. Alright, we talked about HEPA filters. Osmotic pressure, so salt. Remember that even if they have a cell wall, right, you can still, if you have a hypertonic environment, it pulls water out of the bacteria. back in the day game, and the purpose is like you're going across the US in your little wagon cart, and you're figuring out how to preserve all your food, because it's like months. So you would have to salt the meat because you didn't have a fridge. Alright, well, that's, back in the day, people didn't have fridges. Okay, the other thing is radiation. So there's ionizing versus non-ionizing. Ionizing is high energy. Basically, it knocks electrons off of stuff, creates these ions, and then these ions go on to take electrons from other stuff, and it's not good when you're taking it from like DNA or other proteins. So that's how ionizing radiation can kill. This strawberry is already, both of them are dead, right? So here, these that have been radiated, it's dead, it doesn't matter, but you've killed the bacteria on it, so this lasts a lot longer than something that wasn't radiated, the bacteria survived and spoiled the strawberry a lot faster. Non-ionizing is slower wavelengths, lower energy. So UV light is an example of non-ionizing. We will do this lab next week. So essentially what this does is it creates little kinks in the DNA, fuses adjacent nucleotides, and when you're trying to reproduce, those kinks lead to mutations, and it causes all sorts of problems. So this is why you should wear sunscreen. It damages us, damages the bacteria. Okay, and this just sums up everything. Okay, chemical methods. So keep in mind, you affect the cell wall, the cell membrane, the DNA, all that can kill the cell, right? I am not going to ask you specifically how phenol works. I just want you to know what falls under what category, okay? So phenolics are derivatives of phenol. Don't memorize the structure of these things. I'm not going to ever ask you that. Alcohols are alcohol, right? So you can use that as an antiseptic right before you get shot. They can swab the skin with alcohol. Halogens. So halogens. Oh, yeah, okay. All right, we're ignoring this blue part of the periodic table and just counting the columns. So 1, 2, 3, 4, 5, 6, 7, 8. Does anybody know what a valence shell is? Or the octet rule refers to?
Yeah.
There's too many
What is it? The what? The outside. Yeah, so you need 8. So your little element needs 8 electrons to feel happy and feel fulfilled in his life. So in this 8 column, these are referred to as your noble gases. They've got everything they need. They don't need one more electron. They are happy. They are fulfilled. They're living their best life. They don't fight with anybody. So they are very inert compounds. They don't react at all. This column, however, number 7, needs one more, and they're going to fight like hell to get it. So fluorine, chloride, bromide, iodide, I don't know what the last 2 are. But where do we have fluoride in? Toothpaste. Where do we have chlorine? Huh? Pools, yeah. Bleach, right? Bromine and iodine, these are antiseptics, right? So what they do is pull electrons off of the bacteria's structure that end up creating ions that then create more ions, and that kills the bacteria essentially. The other thing that I wanted to point to was this little guy right here. Oxygen needs 2 electrons, and it's going to fight like hell to get it. So oxygen is a very good oxidizing agent, means that it pulls electrons off of things. So it's our terminal electron acceptor, right? It likes pulling electrons and accepting them into its life. Okay. So when I say that I just want you to know what falls under what category, halogens, iodine, chlorine, fluoride, bromine, it's that 7th column, right? And they destroy structures within the bacterial cell by pulling electrons off. Oxygen. toxic forms of oxygen can be fatal, but we're protected because we have catalase, right, enzymes that neutralize it. But if you don't, right, exposure to oxygen ends up killing you. Okay, so we mentioned hydrogen peroxide and we mentioned catalase today. Ozone is another form of oxygen, peracetic acid. We use chlorine, which is a halogen, to disinfect our drinking water. In Europe, they use ozone. Alright, surfactants, think soaps. So these have both hydrophilic and hydrophobic ends. They're good at kind of pulling things apart. So that's what amphipathic means, hydrophilic and hydrophobic. So they're good degerming agents. I'm not going to ask you about this. Alright, if it's got a metal in it, it's a heavy metal. So zinc, silver, all that copper, these are heavy metal ions. It sounds like a metal, it belongs in this category. Alright. Aldehydes is the last part of the name says aldehyde, it's an aldehyde. So formaldehyde is an example. This is something that they use in embalming so that your family can say goodbye and you don't start to smell while you're dying. Gas. Alright, this is what I want to point out. Disadvantages: can be hazardous, highly explosive, extremely poisonous, and potentially carcinogenic. Is this what your go-to? Your first thing you go to clean bacteria off of something? Probably not, right? Alright, enzymes. Lysozyme, this is a enzyme that's secreted in our tears or sweat or saliva. It is an antibacterial enzyme that basically cuts a hole in the cell wall. Now the cell wall is compromised and this guy can die. Alright, chapter 10 deals with antimicrobials, so I will talk about that in a second. Different tests to evaluate how effective these are. Phenol coefficient, just compare it to phenol. If it's better, it's got a greater number. Use dilution, you dip these little cylinders